Hello everyone. Hello everyone, my name is Josef. Welcome to my presentation in which I'm going to talk about life history toxicity on copepods. So why copepods? Copepods are highly abundant in our global oceans. Some even estimate them to have the highest animal biomass on the planet, and they are essential in marine food webs as prey for fish. And this is a picture of the species I've been working on lately. If I talk about life history effects, what is it? Here you see, yes, the life history of the copepod. It develops through, does this work? Yes. 6 nuclear stages, then followed by 6 copepodite stages, and only the adult stage is able to reproduce sexually. And life history effects are effects caused by a stressor, which can be a chemical or something else, which can be, for example, on the development time, or on the growth, or on reproduction, which is illustrated here by a little bit less offspring. And what we observe is that very often we actually see one stressor causing multiple of these effects. For example, in an experiment that I conducted with an antidepressant chemical, I saw that higher concentrations of the chemical lead to a slower development, so animals reach puberty later, but at the same time, once they reach puberty, they produce more offspring. And now I'm really interested actually in the mechanisms leading to those observed effects. And for that, I make use of the dynamic energy budget theory, which is a theory that uses energy translocations in organisms actually to describe the life history. In case of my copepod, it looks like this. Food is used to take up energy, and a constant fraction of that energy is moved to the process of growth and structural maintenance, and another fraction goes into sexual maturation and maturity maintenance. And once a certain maturity threshold is reached, the animal reaches puberty, it stops growing, and starts producing offspring. Here you see a fit of such a model to some experimental data. These are non-stressed data. You see the data points in yellow, and the blue lines are the model predictions. Here, for example, body length over time. And then I also included some temperature-dependent data and food-dependent data, mainly also development times and reproduction data. And I think it's quite a convenient fit, actually. So I tested then the model to explain my toxicity data. So in theory, a toxicant or a stressor can affect any of these processes in the scheme, but I found only one to actually explain the data or the results that I observed, which is actually the maturity threshold value, so the amount of energy that needs to be invested into maturation before it reaches puberty. Because if this value is higher, it means that animals reach puberty later, but they also grow bigger. And if they grow bigger, they eat more and they also produce more offspring. So actually, this mode of action really can explain my data. But to further confirm this, in the next experiment I will test if they also actually grow bigger, which the model predicts, but I have not yet tested. So in a quick summary, DAT models can really help to explain life history effects in a more mechanistic way, like really understanding what is going on. And model simulations can even inspire future experiments, like in my case with the growth experiment that I'm going to do next. And as a little outlook, such data can be very valuable if you want to use models, for example, to extrapolate from individuals to populations. And that's it. Thank you for your attendance.